Our next topic is the hemoglobin. The hemoglobin is present inside the RBC. Students, we have studied the RBCs. So this hemoglobin is basically made up of the two parts, that is heme and globin. So that is why hemoglobin. So there are two parts, heme and globin. The heme part consists 5%. The heme part is a iron containing part, that is Fe containing part, and globin, which is a 95% major part, which is a protein part, means made up of the polypeptides, that is proteinaceous structure. This hemoglobin is a respiratory pigment. We have studied into the respiration. It carries the 4 molecules of the oxygen. So heme is have the iron that is present in the form of Fe2 plus, that is ferrous form, and second one is the porphyrin. Okay students, so there are two parts into the heme part, that is iron and porphyrin. Iron is present in the Fe2 plus form, that is ferrous form. 1 Hb carries the 4 molecules of the oxygen. Now the globin. So globin is made up of the 4 polypeptide chains. So these 4 polypeptide chains are alpha polypeptide, beta polypeptide, gamma polypeptide and delta polypeptide. Okay students, so globin is made up of 4 polypeptide chain, that is alpha, beta, gamma and delta. Now these are the polypeptides, so there should be amino acids. So number of amino acids is also given. Alpha polypeptide is made up of 141 amino acids, beta polypeptide is made up of 146 amino acids, and remaining these gamma and delta are also made up of 146 amino acids. Now on the basis of the polypeptide chains, there are 3 types of hemoglobin are formed in the human body. So on the basis of these polypeptide chains in our body, in human's body, there are 3 types of HB are formed. These 3 types are HbA1, HbA2 and HbF. So HbA1 means the adult hemoglobin. When a human being becomes an adult, the form is HbA1, that is adult hemoglobin, and that adult hemoglobin have 2 alpha and 2 beta, because these globin have 4 polypeptide chains. So it contains 2 alpha and 2 beta, so it will become 4. Then HbA2 means adult hemoglobin 2. So there are 2 types of the hemoglobin into the adult, HbA1, HbA2. It is made up of 2 alpha and 2 delta polypeptide chains. So 2 alpha and 2 delta polypeptide chains are present. And third one is the HbF. HbF means F for the fetal. So fetal hemoglobin. Fetus means when there is a no birth, okay? When which is present inside the uterus of the mother. So when the fetus is present inside the mother's uterus, the fetal HB have 2 alpha and 2 gamma chains. Okay students, 2 alpha and 2 gamma chains. Next, one important point, that is oxygen binding capacity of fetal hemoglobin is higher than the adult hemoglobin. Oxygen binding capacity of the fetal hemoglobin is higher than the adult hemoglobin. This point you have to remember. Then next point, anisocytosis. Now what is the anisocytosis? Anisocytosis means change in the size of the RBC. Students, basically we are studying the RBC, and in RBC the topic, that is the hemoglobin, because the hemoglobin is present inside the RBC. So that is why we have seen the hemoglobin. So anisocytosis means change in the size of the RBC. So there are 2 types, that is macrocytes and microcytes. So you know that macro means larger. So RBC becomes larger than the normal size. That condition is called as macrocytes. That condition is called as macrocytes. And this macrocyte condition is due to the deficiency of vitamin B12. When there is a deficiency of vitamin B12, it causes a macrocytes. Okay, macrocytic anemia. It is called as macrocytic anemia. Then microcytes. Microcytes means RBC becomes smaller, and it occurs into the iron deficiency anemia. It occurs into the iron deficiency because the iron is present into the hemoglobin. Students, Fe2+. So that is the anisocytosis. Next one, lifespan of the RBC. The lifespan of RBC is 120 days. After 120 days, RBC gets destroyed. Where it gets destroyed, we will see in the next point. Then the count. Count is also important. Count, RBC count means the number of RBC in per cubic mm of blood is called as RBC count. So RBC count varies into the male, female and the newborn. So in male, there is 5.5 million RBCs per cubic mm of blood. We are saying the unit is common, that is per cubic mm of blood. And in female, there are 4.5 million RBCs per cubic mm of the blood. So RBCs in female are less than the male. Then newborn baby. In newborn baby, the RBCs are 6.8 million per cubic mm of the blood. Now why RBCs are less in the female? Due to the menstrual cycle, and due to the menstruation, due to the bleeding, there is a loss of the blood to every month, and that's why as compared to the male, in female the RBC count is less. And why in newborn baby the RBC count is high? Because the newborn baby requires more oxygen, because that is a developmental stage, and that's why there is a higher the RBC, that is 6.8 million RBCs per cubic mm of the blood. Now there are some terminologies related with the RBC. Decrease in number of RBC is called as anemia. Actually the decrease in amount of hemoglobin is called as anemia, but you know that the hemoglobin is present inside the RBC, and that's why student, if there is a decrease in number of RBC, obviously decrease in number of hemoglobin, concentration of hemoglobin. So that is why decrease in number of RBC is also called as the anemia. Next terminology, erythropoiesis. Erythros means the red, means red cells. So erythrocytes. So erythropoiesis means formation of RBC. That term is called as erythropoiesis. Erythropoietic organs. Erythropoietic organs means the organs which produces the RBCs. The erythropoietic organs are liver, spleen, red bone marrow. Okay. Next, erythropoietin. What is the erythropoietin? Erythropoietin is a hormone which is secreted by the kidney, and this hormone, that is erythropoietin, stimulates the erythropoiesis formation. That is the erythropoiesis process. Means formation of RBC is stimulated by the erythropoietin which is secreted by the kidney. It is a hormone, that is kidney hormone. Next, the spleen. Spleen is called as graveyard of RBC, because the worn out RBC, or after 120 days the RBCs get destroyed into the spleen. That is why it is called as graveyard of the RBC. Then the spleen is also called as the blood bank of the body. Why? Because it stores the excess RBCs. When the excess RBCs are produced, that excess RBCs are stored into the spleen, students. Next, in adult, RBC produced. Now in adult and into the embryonic life stage, the erythropoietic organs are different. In adult stage, RBC are produced into the red bone marrow. Okay, students, produced into the red bone marrow, and that red bone marrow is present into the, it is a spongy tissue. Then in embryonic life, in embryo, the RBCs are produced inside the liver, spleen and the placenta. So erythropoietic organs are different according to the life stage. Means during the embryonic life stage, the RBCs are produced by the liver, spleen and placenta. But when it become adult, after 18 to 20 years, the RBC are produced by the red bone marrow. So students, here we completed the RBC. We have started the red blood corpuscle, we have seen the plasma and some introductory part, that is vascular connective tissues. So here we completed the RBC, WBC, that is the second cell into the blood. WBC means white blood cell, or it is called as leukocytes. Leukos means white and cytes or cytos means cells. So that is white cells. That is why the another name, that is leukocytes. So the number of leukocytes or WBC is 4,000 to 11,000 per cubic mm of the blood. So these are present into the 1000. As compared to the RBC, the number is very less. Then leukocytosis. What is the leukocytosis? Leukocytosis means increase in number of WBC. Increase in number of WBC means if the WBC increases more than 11,000, that condition is called as leukocytosis. Then leukocytopenia. Leukocytopenia means decrease in number of WBC count, which is called as leukocytopenia. Now another one term, that is the TLC. TLC means total leukocyte count. Okay? TLC means total leukocyte count. Then leukemia. What is a leukemia? Leukemia is a blood cancer. It means that abnormal increase in number of WBC. So in leukemia, the number of WBC becomes more than 1 lakh per cubic mm of the blood. So that condition is called as the blood cancer. So what is a blood cancer? In blood cancer, there is an increase in number of WBC, abnormal increase in number of WBC. Now these WBCs are classified basically into the two groups on the basis of nucleus and nature of the cytoplasm. WBC contains the nucleus, RBC does not contain the nucleus. So there are two types, that is granulocytes and agranulocytes. Granulocytes means the cytoplasm of this WBC have the granules, the particles, which can be stained with a specific dye. These granules show this staining with a specific dye, and the nucleus is lobed into the granulocytes. So there are three types of the granulocytes, that is acidophils, basophils and neutrophils. Acidophils, basophil and neutrophils, these are the types of the granulocytes. Agranulocyte, it means that there is a no granule, that is why agranulocytes. Then the nucleus is not lobed. That is the difference between the granulo and agranulocytes. The example of agranulocytes, or there are two types of agranulocytes, that is monocytes and lymphocytes. Monocytes and lymphocytes. So there are total 5 types of the WBC, that is acidophil, basophil, neutrophil, monocyte, lymphocyte. Out of which acidophil, basophil and neutrophils are the granulocytes, student. You can remember from this. So now we will see one by one this acido, baso, neutro, mono and lympho. So first one is the acidophils. Now why these are called as acidophils? We will see. The size of the acidophil is 10 to 12 micron. The lifespan of the acidophil is 14 hours. Then these acidophils stain with an acidic dye like the eosin. That is why these are called acidophils, because they stain with an acidic dye. That is why acidophils. And another name for acidophil is a eosinophils. Why eosinophil? Because of they stain with a eosin dye, acidic dye. That is why another name is eosinophils. Number, these are 2 to 3% of total TLC. TLC means total leukocyte count. Means out of the total WBC, there are 2 to 3% are acidophils. Then the nucleus is bilobed like this. So this is diagram of the acidophils. The nucleus is bilobed, this and this. There are 2 lobes, and this is the granules which are present, because acidophil is a granulocyte. Now what is the function of this acidophil? This acidophil is produced in the body against the allergy and the parasitic infection. Okay? So when there is an allergy or parasitic infection in our body, the number of acidophil will increases, and that condition is called as eosinophilia, that is increase in number of eosinophils. So students, that is the acidophils. Next one is the basophils. Size is 8 to 10 micron, lifespan is 12 hours, and they stain with a methylene blue. Stain with a methylene blue, and methylene blue is a basic dye, is a base. So that is why this is called as basophils. The number is 0.5 to 1%. These are the minimum. Okay? Basophils are the minimum of the total WBC. They have 2 to 3 lobed S-shaped nucleus. Then they secrete, the function is they secrete the heparin. Heparin acts as an anticoagulant, natural anticoagulant in our blood. Then histamine. Histamine is present in allergic reaction. It gives a response in allergic reaction. And then the serotonin. Serotonin is a chemical messenger. This serotonin involves in the messaging or to increase the motility of the intestines or GI tract. Serotonin increases the motility. So this is the function of the basophils, and it have 2 to 3 lobes. So here I am showing 3 lobes, 1, 2 and 3, and it looks like a S shape, and these are the granules. So this cell is the basophil. Now the next one is the neutrophils. The size is 10 to 12 micron. The lifespan is 12 hours, and they stain with a neutral dye. That is why these are called as neutrophils, or they doesn't stain. The percentage number is 60 to 65%. So neutrophils are the most abundant WBC. These are the maximum WBC. Okay, neutrophils. Then the nucleus is 3 to 5 lobed. So here I am showing 4 lobes. 1, 2, 3, 4, and this is the granule. So there are 4. So nucleus is 3 to 5 lobed, and function is these neutrophils are phagocytic in nature. These neutrophils are phagocytic in nature, means the neutrophils engulf, phagocytize the bacteria and the viruses. Okay, students. Next one is the monocytes. The monocytes are 12 to 20 micron in size. The lifespan is almost 20 hours or less than 24 hours. The percentage or the number is 6 to 8%. The shape of the nucleus is kidney shape or bean shape, like this. The kidney shape, or you can call it as a bean shape. Then the function of monocytes. Monocytes acts as the scavengers of blood, means these monocytes engulf damaged or dead blood corpuscles, dead blood cells. That is why these are called as the scavengers, which engulf the blood cells which are damaged or dead for the purpose of cleaning from the blood. So students, here we are seeing the 4, that is acidophils, basophils, neutrophils and monocytes. Our next will be the lymphocyte. Next type of the WBC is the lymphocytes, and this is the last type, that is which is included into the granulocytes. The size is 6 to 16 micron. Then the lifespan is 5 to 7 days. Then the nucleus is a large oval shaped nucleus is present into the lymphocytes. Then the count, these are 20 to 25% of the total leukocyte count, that is TLC. Types, there are 2 types of the lymphocytes, that is B lymphocyte and T lymphocytes. Now the name of B lymphocyte and T lymphocyte is derived from the lymphocytes which gets mature into the bone marrow. Those lymphocytes are called as the B lymphocytes, and the lymphocytes which mature into the thymus gland, those are called as the T lymphocytes. So this B lymphocyte produces the antibodies, and this antibody provides the immunity which is called as the humoral immunity, because these antibodies are circulating in the blood. Then T lymphocytes again provides a cell mediated immunity. So these T lymphocytes again have different types, that is the killer T cell, helper T cells, which kills to the foreign body, maybe the bacteria and the viruses. So these lymphocytes basically involve into the immune system. Okay students, so production of antibody is the function of B lymphocytes. So here we completed the WBC, that is white blood cell or the leukocyte. Now the next topic is the platelets. The third component of the blood cells or blood corpuscles is the platelets. The size of the platelets is 2 to 3 micron. The lifespan of platelet is 2 to 4 days only, and count is 1.5 lakh to 3.5 lakh per cubic mm of the blood. So remember the count of the RBC, WBC and the platelets. Then platelets are also known as thrombocytes, because these cells helps in the or causes the thrombus formation, that is the clot formation, that is thrombocytes. The platelets are derived from the megakaryocyte cells of the bone marrow. Megakaryocyte cells are the cells of the bone marrow. Okay students, and from these megakaryocytes there is a formation of the platelets. So platelets are the fragments actually of the megakaryocyte. Shape of the platelets, the shape may be disc shape, may be oval shape or the biconvex shape. Remember RBC is a biconcave and platelets are the biconvex. Then there is a one term, that is thrombocytopenia. What is the thrombocytopenia? Thrombocytopenia means decrease in number of the platelets. So you will observe this thrombocytopenia in a disease, that is dengue. So in the person which is suffering from dengue, their platelet counts decreases. Dengue is a viral disease, so there is a decrease in the platelets, so you will observe thrombocytopenia in a dengue disease. Function of the platelets. Platelets repair the endothelium of the blood vessels. Endothelium means it is the inner lining. Suppose this is the blood vessel, maybe artery or maybe veins. So it have the inner layer, and if this inner layer gets injured, so the repairing of this endothelium is done by the platelets. Then these platelets synthesize the thromboplastin. This thromboplastin helps in the clotting process. We will see the next topic, that is the blood clotting or coagulation of the blood. So this is the platelet, students. So here we completed the blood. We have seen the plasma, and plasma is 55%, and the blood cells or blood corpuscles which are 45%. So in blood corpuscle we have studied the three types of the cells, that is the RBC, WBC and the platelets. Now our next topic, students, is the blood clotting, or you can call it as a coagulation of the blood. So blood flows from cut or wound, but after some times it stops automatically. Students, you have observed, or if you have noticed, when you have got an injury, that is minor injuries, if there is a cut or there is an abrasion, if you are fall, so there is an abrasion, so there is a bleeding, and if you have noticed that bleeding after the sometimes means within the 5 to 10 minutes, that bleeding is automatically stops. Okay, so that is the mechanism of coagulation of blood. So naturally our body prevents the loss of the blood from that wound or from that cut. So that is the blood clotting. So blood flows from cut or wound, but after some times it stops automatically. It is called clotting of the blood, and that is called as the clotting of the blood. Now the timing, the normal clotting timing in a healthy person is 2 to 8 minutes. So generally the clotting process takes 2 to 8 minutes. So the bleeding will be stopped automatically within 2 to 8 minutes. Sometimes clots are formed in the intact blood vessels, which are of two types. Now it means that intact blood vessels means no ruptured, and here is a rupture of the blood vessels. So intact means no cut, this is a continuously, or cut or wound means the blood vessel get ruptures, but intact means continuous, there is a no breakage. So sometimes the clotting also occurs into this intact blood vessels, and which are of two types, that is thrombus clot and the embolus clot. There are two types, that is thrombus clot and the embolus clot. Thrombus clot means it is a static clots which grow bigger and bigger and ultimately block the blood vessel. So these are static, stagnant, they does not move with the flow of the blood. That is the thrombus clot, means which grow bigger and bigger. So it will grow and it completely blocks this blood vessel. That is the thrombus clot. If such a thrombus clot is formed in the coronary vessels are the vessels of the heart, that is the coronary arteries. So if this clot is formed in the coronary vessels, then called as a coronary thrombus, because that thrombus is formed into the coronary vessels, and that can cause the heart attack, because due to obstruction in the blood flow, due to formation of this thrombus, the blood supply to the heart muscles will be stopped, and then cells start to die. That condition is called as the heart attack. If such a thrombus is reached into the brain, it is called as cephalous thrombus, and it can cause as the paralysis, because the muscles are controlled by the brain, and if the part of the brain is damaged, then the loss will be the control of the brain or the muscle will be lost, which that condition is the paralysis. So this is a very serious condition, that is the clots, thrombus clots. Then next one is the embolus clot. Embolus clot means the moving clots which flow with the blood. Means it is a moving, it is not a static. Such a clots are called as the embolus clots. These are more harmful due to the moving nature, because it can reaches from one place to the another place. They can reach into the coronary vessels, they may reach into the your brain, so it can damages to the brain and the heart muscles and your important systems. So students, here we completed the introductory part of the blood clotting. The next topic is the clotting process, how the clotting occurs, which are the enzymes and the chemicals are involved in the clotting process.